our introduction. Welcome everyone to the Mowat-Wilson Syndrome Foundation Meet the Researcher webinar series. Today we're very pleased to have Sumantra Chatterjee with us, and we just appreciate all of you coming today and learning about his research and learning more about all the different researchers that are working on MWS research. This is our final webinar series for the year of 2021, but we are already getting researchers lined up to start again in 2022. So we'll take a little break and have more in the series next spring. Again, I want to remind everyone that today's session is being recorded. So my name is Marcy Smith, and I am the Executive Director of the Mowat-Wilson Syndrome Foundation. The mission of the Mowat-Wilson Syndrome Foundation is to enhance the lives of people affected by Mowat-Wilson syndrome by providing family support, raising awareness, and supporting research and education. And we appreciate you coming today. Today we have Sumantra Chatterjee. He is a research assistant professor at the Center for Human Genetics and Genomics in the New York University Grossman School of Medicine in New York. Sumantra has trained both as a developmental biologist and as a human geneticist studying complex congenital diseases. His work on Herschsprung disease has uncovered how multiple genes and mutations are interconnected, which has helped to explain the severity of the disease observed in various patients and also helped to classify patients better. Before we get started, I just want to let everyone know how we'll proceed today. In just a moment, we're going to let Sumantra talk about his research. When he's finished, we will take questions in the chat box. And so we'll have, you can ask questions there. And I'm going to ask that you keep your questions to the end of his presentation. So again, I want to thank you all for joining us today. Sumantra, I really want to thank you for all you've done and for your research. And now I am going to let you take over. Thank you, Sumantra.
Thank you, Marcy, for that lovely introduction, and also to the Mowat-Wilson Foundation for this kind invitation. I really look forward to this talk, and I hope I can sort of engage more people in this audience to ensure that research in this kind of rare disorders go forward. And I promised in the last year's presentation, I'm for the long haul. I will never desert this. I will be an ally on your side. I would start sharing my screen, so just let me know if you can see it. If you can see it, you can see the slide. Excellent. There's no Zoom problem then. So as Marcy said, I'm a research assistant professor at the NYU School of Medicine in the Center for Human Genetics and Genomics, as well as I have an affiliation with the Department of Neuroscience. So I straddle both basic biology of neuroscience as well as the genetics of human diseases. And my work in the last decade or so has sort of more and more focused on congenital disorders where kids are born with, but as you know, they're much rarer, and efforts to find the genetic causes has not been or the phenotypic causes has not been as severe as many other disorders people have worked on. And as I said, I'm fascinated, and I'm sort of hopeful that as we go forward, more and more people will join our journey. And this rare is not unimportant would become a mantra, that just because it's rare, we don't have to neglect it. There are people who really need the answer to this. So today, what I will do is I will try to connect my current work, which I work on another congenital disorder, but which many of you might be aware of, on Hirschsprung disease, with how we plan to use the tools and techniques and knowledge we have gleaned from studying that to Mowat-Wilson syndrome. And primarily both from a research perspective of trying to understand the biology, as well as from a patient perspective of trying to understand better the genetic consequences of Hirschsprung disease and Mowat-Wilson syndrome together. And I will give you a brief overview of what I have done, what I plan to do. And then as Marcy said, I will be more than happy to answer questions of like how I can improve this, any other suggestions you have. Maybe I should, I'm sorry, I should go to slide format. Oh, there you go. So I thought what I will do is first sort of give you a brief outline of my life, my journey, to get you on the page of why me, why not somebody else doing this, or how can you trust me that this guy knows anything he's doing. So as you can make out from my name or from my accent, I originally am from India. I did my basic undergraduate training in India before I moved to Singapore to do my PhD. My PhD was on mouse genetics. There was no human development, anything human disease involved in it. It was a pure classical training in trying to make animal models and trying to understand what genes do in development, which I didn't realize that time would play a large role in my later career for looking at diseases of congenital diseases of birth, because I worked on mouse genetics of development, which you can now translate into things. But while I finished my PhD, I'm an impatient kind of a person. I wanted to learn something new, do something better. I decided to sort of transition, if you may, into looking at human diseases and understanding human genetics. So I made the 10,000-mile journey across multiple oceans to the United States. I came to Baltimore at Johns Hopkins University to train under Aravinda Chakravarti, who's a leading human geneticist. His real lab and his research worked on Hirschsprung disease. That's where I first heard about it. And I will admit to it, I'd never heard about the disease before I met him. So to me, this was an eye-opening world. I've never dealt with patients. I've never dealt with patient DNA before I joined his lab. All my life, I worked with mice. So joining his lab and the training with him in Baltimore was really, really instrumental in me, one, understanding the practice of human genetics, if you may, but also getting me hands-on into trying to understand a complex disease where many people were not sort of putting focus on in Herschbrand disease. The recent, the last 3 years, we've moved to New York, where since last year, I've been running my own lab in the center for looking at these kind of connections and complex diseases. So my journey has sort of straddled both fields. I still work on the basic biology of development, but I have more and more lab is transitioning into using that knowledge we gleaned from understanding the basic biology into explaining, if I may use the word, of the diseases of congenital disease, the disease of nervous system in children. Right. So as I mentioned multiple times, I work on Hirschsprung disease, which maybe to this audience is not unknown, but I've realized around the world sometimes when I go and give talk, it is a disease which is not really well heard of, and people sort of look at me blankly, wondering if I made this up. But as I said, this audience is educated enough, and I'm sure you've come across this. It's a disease of the gut where kids are born with almost reduced or no ganglions in your gut. And also this is kind of a knowledge base where I have to explain to people that beyond your brain, there are also nerves in your gut, because whenever you think of nerves, you think of the brain, but your stomach has 400 to 600 million neurons, and they do as much diverse function as your brain does. It does everything. It does every same type of neurons your brain has. So the fact that it is that complex tells you that it requires a regulation like your brain. So to understand it, we require to have the same tools and same depth of experimental expertise to get to that understanding why this disease occurs. This disease is not very rare, in spite of what people think. Now the number seems to be even higher, at least in European ancestry populations or whites, it's in 1 in 5,000 live births. In Asians, it's almost double. Asians get about 1 in 2,500. The numbers in Africa is still a little bit underrepresented and understudied, but our recent forays into looking at epidemiological data suggest the numbers that might be even higher in African Americans, and so the numbers in Europeans. So and this is where places like the Motor Wilson Syndrome Foundation plays a very important role in collecting numbers, because first starting anywhere in any disease to know how many people it affects. And so this number, as I said, this number is not as rare as we initially thought it to be. One very interesting aspect of this disease, at least for Hirschsprung, it's a male disorder. So there's almost a 4:1 bias. So you get 4:1 more male childs get affected than females do. And females do get affected, it's not zero. But sometimes when females get affected, the female affection is much severe, because the disease can be manifest in 3 different forms. They're called short segment, where your neurons are only missing in the very distal part of your large intestine, near the anal end. It can be long segment, where much more distant, much more of the colon is affected. Or you can get something called total colonic aganglionosis, where the whole large intestine has no gut, which is very severe, but it's extremely rare, as you can make out. And female affected, we have found out now with numbers, seem to be more affected in the long segment, the TCA categories, where females, when they do get the disease, they get a severe form of the disease. whereas males generally get the milder form of the disease. And over the years, and this is not my credit, many labs have worked on it over the last 40 years now, including Aravind Chakravarti's lab. We have now at least identified, unlike Ward-Wilson syndrome, there are at least more than 24 genes where patients have mutation which we can connect to the disease. For all of them, we don't know why it causes the disease, but clearly compared to a control population, that mutation seems to be more in the patients than in an unaffected population, which sort of tells you right off the bat that this sort of understanding this requires a little bit more tweaking, because now we have to connect 24 genes to this phenomena, because individual genes don't get it. But also it's interesting for this disease, at least, is that along with genes itself, there are pieces of DNA in the genome called enhancers, which control how much the gene is turned on and turned off. So we have found patients, many patients actually, where there are mutations in those lights, the switches. So explaining the genetic change is easy, explaining this is even more harder, because it's like if you have a genetic change, it's like if I switch off, break the light bulb, that's why you get darkness. But a switch is like a dimmer switch. You don't break the switch. You basically turn it off and turn it on. So understanding what that mutation do, how does it affect the turning on and turning off of the gene, has been one of our sort of insights of finding in for these kind of diseases. But before I sort of jump and say, oh, there are 24 genes and 50 genes and 100 things we found out, let me also be clear. We have now discovered that in majority of our patients, about 60-70% of our patients we have sequenced the two genes which keep turning up again and again. There's one gene called RET and the other gene called EDNRB. So both of these are receptors on the cell surface of these neurons, the gut neurons, and the signal. So we think that the disruption of that signaling causes these neurons, nerve cells to start dying off or not traversing through the gut and not colonizing the gut. So sort of in a way that was good, because it gave us a window into understanding what this disease is. Like in Ward-Wilson, you have a single gene ZEP2. Here at least it gave us two genes to sort of start understanding the remaining 24 genes. But to have a better understanding of this, as I said, though these two genes are important, it's not the complete story. A couple of years ago, we completed a sequencing run of 190 individual cases we had, who had all of them were short segment Hirschsprung disease. We knowingly did one type of it is to keep the experiment simple, because if you put in too many variables in the experiment, gene discovery sometimes becomes difficult. We can't have enough power to detect genes. So we restricted our search to these 190 cases who had all short segment, and they're all individual cases. That means they had only one member of the family who had the disease. There was no other afflicted member, neither the parents or siblings or cousins. So these are short segment isolated cases. I mean, this is it's an experimental tool to keep your experiments focused. So the discovery of a rare disease becomes more stronger. The moment you throw in multiple types of variations initially, your power to detect something becomes weaker. So it's our way in to first understand. But what was remarkable for us is once we started finding these genes with just 190 patients, of course, we found RET and EDNRB to our not surprise. Thankfully, it's kind of way we discover same things we discovered. But we also found, if you look at the top right column, we found genes which help RET to express. So GDNF is a ligand. It's a thing which binds to RET for signaling. So we found patients who have defects in the other signaling pathway, which controls RET. We found the signaling molecules of EDNRB also had mutations. So the patients who had a mutation in the pathway which controlled EDNRB. We had genes which were what are called transcription factors. These genes are what control other genes. So you not only have a mutation in a gene, you can also have a mutation in a gene which controls that gene. So any of these ways you can sort of detect to change the expression of a majority of these genes in the gut and sort of disrupt the system. The system needs to be disrupted for the neurons not to grow. And we found out these genes are not unrelated, because we used to find these genes one by one in patients. As we started putting this picture together, it's like a puzzle. Suddenly the corners started making sense. You have SOX10 mutation because SOX10 is a gene which controls RET expression. Hence it explained the connection. So it was not unrelated. So just because we found them unrelated, biology is not unrelated. Biology has a logic which we are discovering as scientists. And these kind of experiments, sequencing patients really help us to get that logic, because in a rare disease, a lot of people will not have SOX10 mutation. But if you find two patients with SOX10 mutation, at least you know that mutation exists, you can connect it now to RET. So the more we increase number, the more these kind of things fall off and the blurry puzzle starts getting more clear in our head. But we can do this now. Technology is there. It's called exome sequencing. It's no longer like a rarity. People are doing it as clinical tests as well in many, many conditions. Right. This is one of my academic slides, and I apologize in advance if it's too crowded, and I'm more than happy to explain in detail later on. So as I told you, we in Hirschsprung disease, surprisingly or not surprisingly, we also find mutations in the enhancer, the switches, which is not the gene, the switches which control. So the schematic or the cartoon at the top, so as you can see this thing called red and black, that's a gene. And in red, I have marked out portions of the region of the red, which are switches, which we know. And patients have mutations. So for example, anything in red is a patient mutation. Anything in black is what normal people, or I'm sure say now, people who don't have the disease have it. And so it was always a mystery to us. How does that change? Why do a thing which is not part of a gene, why would that mutation cause a disease? It turns out these pieces of DNA can control the expression of the gene. So if you make these small changes on the bottom left-hand corner, I've drawn out. So these numbers are basically the green things are the same region as the region of the red at top. And this red bar here basically tells you that we have made a small mutation which mimics a mutation in the patient. And on the right graph, we are basically measuring how much red gene is produced, right? So one means it's produced completely normally, right? And as you start making this mutation, you can see the expression of the gene falls. So it explains why that mutation, the change in that non-coding DNA leads to a disease, because it doesn't require, the gene is perfectly normal. The switch is broken. And which in turns leads the gene expression to fall, and the neurons don't see the gene much, and the neurons don't know what to do with it, and they start dying, right? But what was more exciting to us was we always in the lab initially used to study these pieces of DNA one at a time. Remember something, human beings are not one at a time. A human being has all these mutations, or none of the mutations or combinations thereof, right? I mean, we study them as one at a time, and then we sort of add it up. The question is, now we have technologies to delete all of them together or make the same mutation which the patient has. So that's what we did. That's the bottom right two I'm showing you, which says 3 mutations together or 4 mutations together. And what was really satisfying for us to see was when you had 3 mutations together, expression of the gene falls significantly, almost 25%, right? And this number here called OR means odds ratio. This basically means what's the chance of a patient getting a disorder if you have this combination of disruption. So 1 point, this basically means 1 basically means you have no chance of getting a disease. 1.6 means it's almost 60% more chance of you getting a disease than a normal background person who doesn't have any mutation. When you mutate all 4 enhancers, the chance of disease is 12-fold. So compared to a patient, compared to a person who has no mutations, if you have 4 mutations, you get 12 times more the disease. And it always surprised us. The reason is this, because when you get all 4 mutations, expression of the gene falls to 50% of normal. So now think about it. If I reduce your salary by 50%, you will find out. Similarly, if I reduce a gene expression by 50% in a cell, the cell is detrimental to the cell. Hence, you can now connect this genotypic consequence to a phenotypic consequence. You get the disease that high because the gene expression is extremely low. And this is to me very, very satisfying, because for the first time in a long time, you can now go from a patient data sequencing to actual functional consequence of that change. And we can do it now routinely for many such things, and to understand why certain disorders or certain mutations will have a more severe effect than others. And I would really be, hopefully before I finish and I can see a day when you give me a sequence and I can predict or explain to you what the physical consequence would be. We are far from there, but at least we're taking the baby steps. A journey of 1,000 miles begins with 1 step. I'm hopeful we're taking those baby steps together. And one day we'll do this. And so no patient left behind. We can explain everybody, and we can give you some sort of an idea of what it does. So I'm this kind of thing satisfies me, though it's an academic study, and I apologize, this is too much information, but this is what it does. The other thing I told you is, like, ultimately it affects the gut. And unfortunately for congenital diseases, getting human fetal tissue is very, very complicated and it's difficult, and I appreciate that. But that's where my training as a developmental biologist in mouse helps, because now we can model this disease. We can model these diseases in an animal model where you have much less restriction of collecting tissue at the correct time point. This disease occurs during development in the womb. So we can get mice at the correct stage when the neurons are migrating through the stomach, and we can dissociate these cells and see what exactly happens in those cells when you have this knockout. So that's what we have. We made a mice where we have knocked out red, which we know is as told is a major gene for red. And now what we can do is, instead of taking the whole tissue, we can exactly take out the cells required. The technology in the last 2 years have really sort of exploded. We can now study a single cell. We can see what happens in cell. So each of this colored dot in this like fanciful looking graph is nothing but a single cell. And it has been measured for expression of all its genes, which we can measure. And based on that, we can cluster these cells. We can say cells which are all green in color appear to do a similar function because the gene expression looks exactly the same, right? So we've taken it out and we clustered it back. And we can now we can ask the question, what happens when you knock out red? Because that's a major phenotype in human patients, the red deficiency. When you do that, two things appear to us. First of all, for the longest time, it was assumed red is only expressed in neurons. And we went along with that. We said, that's fine, that's what it could be in the gut. It turns out when it is in the mice, so these orange cells at the top are not neurons, they're glial cells, they're supporting cells of neuron. It turns out red expression is also it's sort of predominantly expressed in the supporting cells. So when you have a deficiency of a gene, it not only affects the neurons, it affects many other cell types. So the severity of the mutation of red patients is simply because the expression of red is much more ubiquitous than we initially thought it to be. But what we can also do, and I'm not going to bore you with all the details of like, what this red means and green means, but we can now classify what class of neurons these are. Because just to say tell a patient that, oh, by the way, your neurons are missing is not very informative. It appears all neurons are not missing. Even when you have a mutation, only a subset of neurons are missing. Right? Even when the most severe of cases, red deficiency only leads to a subset of neurons dying. A subset of neurons still survive and they thrive. So basically, the patient is not completely aganglionotic. The patient has some ganglions, right, for the mouse model. But what we can also do is, and on the right graph is basically the same graph. The darker color you see means the gene is expressed higher than the greener color. So I took all genes which we know have human patient mutations in Hirschsprung disease and asked the question, where do they express in these cells of the gut? As you can see, there's a varied distribution. So this assumption is that, oh, I have a mutation in SOX10 and I have a mutation in EDNRB will give me the same phenotype is wrong. Because EDNRB and SOX10, for example, these two genes express slightly differently. RAR beta express slightly differently. So you can now sort of start predicting what the mutation will wipe out or affect what cell type. So this is kind of, again, another way of doing a genotype to phenotype correlation. So if a patient comes in with a specific mutation, you can explain a red mutation will have much more severe because you can see in the top left-hand corner, red expresses the darker color in almost majority of cells. Whereas EDNRB is only in one corner of these cells here. So you can now classify cells by expression and by mutation. So you can predict, and these are of course mouse studies, but hopefully you can predict that going forward, if you have a patient with a specific mutation in a specific gene, you can come in and say, this potentially are the group of cells which we're missing, the severity would be this. So this is what we can predict would happen to a child who has that mutation versus somebody who has a much more broader mutation, who might have a much more broader phenotype, and which might reflect, and you can explain that phenotype even if you can't do anything right now about it. And this, I think, is going to be very powerful tool going forward, especially because for congenital diseases, as you understand, you are born with it. So we can never go back in time in human tissue, but we can go back in time in mice. And we can use that to extrapolate to humans and say, this predicted what happened in the humans. Right. Again, I will not take too much time. And we can also, these technologies can also give us gene discovery. For example, in the bottom left-hand corner, we found this gene called DBH. It's a neuronal marker for differentiation, which was unknown to be expressed in the gut. We found that some cells which express red also express DBH, and it helps differentiate the neuron down a specific pathway. So some of these motility disorders we see of humans can be explained by certain specific genes which are expressed or not expressed due to these kind of changes. And we can, as I said, I'm just trying to sort of let you know that we have these tools now, and eventually we would use these tools for gene discovery in human tissues, even if it's limited numbers we get. Anyway, as I said in the beginning, the discovery is also not about just finding random genes and cells and sort of move on in life and get to the next project. The aim is to understand, why do we get these genes? There are 23,000 genes in our genome, in the human genome, right? In Herschbrand, there are only 24, 25 genes affected. So why those 24? What's so special about those 24, right? There's nothing to suggest why not remaining 22,973 genes. There's nothing special about them, right? It turns out, as I said in the beginning, maybe I gave my hand away, these genes are not interconnected. These genes are not unconnected. These genes are interconnected to each other by the way they regulate. So now we have found, so every gene marked here in red is gene where a human patient has a mutation, which leads to Hirschsprung disease. We found out that these genes are not unconnected. Red controls its own ligand, GDNF, and its receptor. These things at the top are transcription factor, which controls, I told you, via binding to the enhancer elements, controls red and EDNRB. EDNRB, of course, is a gene which is in the same cell surface as red. So all of these, a neuron or a cell in the gut sees all of these genes in like functioning together. You perturb any one of these, it perturbs many other things in it. Like it's like a family that's slightly dysfunctional. So one member goes a little slightly crazy, everybody's affected. Or I like to say it's like your neighbors. You might not like your neighbors, but you live with them. So if one of your neighbor makes a lot of noise at night, your sleep gets affected. So you don't need mutations in many, many genes. Any one of these mutations, any of these genes will perturb this whole network. And the cells then sort of start seeing this perturbation and sort of responding to that perturbation by either dying or changing fate or not becoming the things they want to become. And I will sort of end this part by giving one sort of, again, things of my, I enjoy my, these are days I enjoy my work really, really very, very much. So we found this gene at the top left-hand corner called RAR beta just by functional studies. There was no known human patient mutations in Hirschsprung disease for this gene. We found this gene, we knocked it out in a mouse, we showed it leads in the mouse to same kind of phenotype, they have controls red, and everybody challenged us and said, that's all great, but if there's no human patient with that mutation, it doesn't make sense. It just, it's an artifact of your experimental system. And we're like, well, that's fine. I agree, I admit, I have no answer to that. 2 years after we sort of finished the study and sort of trying to explain this, a patient with RAR beta mutation was sequenced, and he had found RAR beta mutation, and he had constipation questions. Initially, the doctors did not really pay attention to that because this young woman had many other phenotypes. So the constipation was sort of the least of the worries for the parents. But after the publication of our paper, the clinicians and the sites went back to this family and requested if they can take a rectal biopsy. As you can imagine, being parents yourself, sort of consenting for rectal biopsy is not the easiest on your child. But I think they had faith and they did go and take rectal biopsy. It turns out the kid also had hypoganglionosis or loss of entry neurons. So the gene was completely unknown before doing this. We found just by functional screen, and this at least will help explain one of the phenotype in that young woman. And I said, these are the days I really enjoy my job. I mean, for every frustrating day we have on the lab, these kind of single discoveries and just sort of helping people. It's, I mean, at least getting a family know this also is a thing. It's really, really satisfying to me. For even if it's n is equal to 1, as I said, these are the days I live for. I get out of bed every morning, hoping the work can help others. Right. So what has it got to do with Mort-Wilson syndrome? You might be wondering, this guy is yapping on about like Hirschsprung and this and Hirschsprung and that, and he can take cells. So I came into Mort-Wilson syndrome via 2 ways, I will tell you frankly. And the first sort of, because as you might be aware, and this is not the audience to sort of teach this, that 50% of Mort-Wilson syndrome cases also get Hirschsprung disease. So it's called a syndromic disorder because we also can get Hirschsprung disease, and it was called Hirschsprung disease with neurodevelopmental disorders. The early name of Mort-Wilson was called Hirschsprung disease with. So Hirschsprung disease was a way in. And so when I started Hirschsprung disease, I also thought about other disorders which come with Hirschsprung disease. One of them is Mort-Wilson. There's also something called CCHS. There's L1-CAM syndrome. There are many disorders, familial dysautonomia, where there is Hirschsprung disease is an associated syndrome with the disorder. So that was always sort of my peripheral vision, this disease, and it always intrigued me. There's a transcription factor, ZEP2. As I said, I showed you SOX10, and we found other transcription factors. It always fascinated me. But my real introduction to this, or my real interest in this, was COVID. It was 3 years back now. I gave a talk at the American Society of Human Genetics meeting, and to my great fortune, Dr. David Mort was in the audience. And after I gave the talk and I talked about this interaction in networks and transcription factors, and I had never met him in my life, this gentleman came up to me, and very polite, very soft-spoken gentleman, and he told me, "Oh, I'm really interested in like Mort-Wilson syndrome and I did ZEP2." And I was thinking to myself, "That's good." I was like, so, and then he introduced me himself, and I was like, "Oh, this is David Mort." And he said, "Would you be interested in looking at some of these?" And I said, "Sure." I said, I mean, I'm always thinking on this about other disorders. So that was my introduction to it. And then I really seriously in 2019, I thought about sort of foraging into away from looking at what happens to these kind of patients with Hirschsprung disease and why other disorders get it. And I told you in the beginning, we study diseases in isolation. Like, lab A studies this disease, lab B studies this disease. The body doesn't care. The human biology doesn't care if Sumantra can only study Hirschsprung. The diseases occur simultaneously sometimes. So it was fascinating to me. Why does 50% patients only get Hirschsprung disease? Because uh, the conventional wisdom was where the different kinds of mutation leads to different kind of phenotypic outcome. Though that might have been true, the more I read about it, the more I realized that's not completely the picture, because there is no correlation from what kind of mutations you have in the gene ZEP2 versus what type of disease of Mort Wilson you get. And plus, as you would know this better than me, Mort Wilson has multiple phenotypes. You get effects in the brain, you get effects in the other tissues, the heart. So it cannot purely be the mutation itself. So it suggested to me that two things happening. There might be other genes which, along with ZEP2, regulate these things and things. You need ZEP2 and plus something, and you also might and also the way ZEP2 is affected might affect what other tissue they're doing it. So I told Dr. Moore, what I can do is I can look at this connection and try to find out, is the ZEP2 connection relevant in the brain versus, let's say, the gut. And as I said, they're two different neurons, but are the same basically. We're just studying them differently. So that's how I got it. So this immediate focus, what I want to really do was to understand the genetic causes of variability. Like, is it truly, how many other genes might be playing a role along with ZEP2 to do this? And to do that, we require patients who have different kinds of effects. Effect of the brain, as well as either Hirschsprung or not Hirschsprung, because that will then sort of help us distinguish the patient population. I'm sorry, I will stop you. I think the light went off. Okay, give me a couple of minutes. I'm really sorry for the apologize for that. I'll turn on the light. Thank you. I'm sorry, the joys of too much modernity. It turns off the light off, you don't move too long. So power saving is good, but it's also, I apologize again. So my way in this was, the more I read about it in the last year or so, a lot of work has been done, the groups in Italy and all, where they've actually done MRI scans in the brains of Mort Wilson patients. They've found proper, like, defined defects of in the brain, especially in the hippocampus. And as I said, I was completely unaware of this. This was my reading into it, and that told me that it's not just when you say, oh, there's a brain defect or intellectual disability. It's disrespectful to the patient, it's disrespectful to the community to broadly classify and say, oh, the patient has mental ill health issues or intellectual disability. The brain doesn't work that way. There has to be something specific. And I realize there's already some work done, and people know there's parts of the brain which are affected in Mort Wilson patients. The sample size is not very big, as you can imagine, but still it gives you a way. And they have Hirschsprung, which tells you that there is clearly two nervous systems, your gut and your brain, which are somehow connected. There's something they're doing which is like affecting some of the kids with both the disorders or both parts of the disorders. So that, from a biologist perspective, was very interesting to me. That can we study neurons in that sense as a disease which has both affected neurons, what they do, and why only 50% cases have it. And there's a family in Italy in this paper which is cited at the bottom, where they found the 6 members of a family at Mort Wilson with almost identical mutation they detected, but they're very different phenotypes. So that's this, to me, it cannot be the same mutation. If the same mutation cannot have 5 different expression in the human child, right? So that basically means we're missing something. So then I came up with a plan, and this I presented, I think, last in the meeting of the syndrome, the foundation meeting, is that how can we better understand this? And with my expertise in Hirschsprung disease and building mouse model and looking at human genetics, how the best way to do it. And the plan I came up with is, like, we collect enough patients of either Mort Wilson alone or Mort Wilson with Hirschsprung disease, because these are 2 classes of cases which we can explain the phenotypic difference. We do sequence the whole patients, and we ask, beyond ZEP2, what else do we find, especially in patients who have Hirschsprung disease. So, and if you can find that, that's a way into finding out other genes which can explain the phenotypic variability, especially for the gut phenotypes. And parallelly, we also try to recreate these animal models. We make the animal models of ZEP2 and Rett and see, can we recreate this phenotypic variability we see in patients? Because in these cases, as I showed you for Hirschsprung, we can take out cells, we can take out tissues, we can genotype them, we can make different kind of combinations and see, can we increase severity as we increase sort of the genetic defect in these mice. So to compare these two, we sort of hopefully come up with a global picture of what happens in patients where you get a severe kind of Mort Wilson with another disorder versus where you can just get Mort Wilson or just get Hirschsprung disease. So that was a broad plan. So I drew up a flowchart last year, and this was in collaboration with Dr. Morton Wilson and Dr. Margaret Adams in Seattle. So sort of the idea is in human genetic study for right reasons, patient consent is very, very important. You don't want to just give me random stuff and trust me. So we sort of thought one way of doing it is having a good uniform patient consent. So all of you know exactly what you're signing up for, you know what it is, and the foundation plays an important role in collecting it. So you are in charge of your own samples and data, and you know the phenotype. From a research perspective, all we will get from you is de-identified samples. All I need to know is that it's the affliction status, like does it have only a Mort Wilson? And if you have parental DNA, the parental affliction status, and most importantly, sex or gender, which we can find out from sequencing. Beyond that, I don't really need to know anything. I mean, you are fully in charge of the data. The clinicians are in charge of the data because we just need the DNA to sequence it. We sequence a whole genome to find any changes. We figure out additional mutations, and we try to then connect this mutation to the phenotype. As I said, this data would be completed into the foundation. I mean, as you guys start building servers, they're large data sets, but you would be in charge of your own data. You can actually see. I am not a clinician, so I'm not in authority to either return results or interpret in terms of the clinical result. But I'm sure Dr. Wilson and Mort and Dr. Adam, they are much more organized in this and they know this. So you would be completely in charge of your data. I mean, let me make it very, very clear to the foundation and to everybody. This is, you will not see me taking and running away. And this, I know that we scientists sometimes get a bad rap for this, for not really being sort of, not being sharing data, but this data would be completely yours. The only thing we need to do, because if you're going NIH funding, if you get funding from private institution, that is not a requirement. If you get NIH funding, the raw data has to be uploaded in a public database because it's a public money. It comes from you and my taxpayer money. But again, this is a de-identified data. So there's no way you can find out. You will have a number, which is a random number. And so there's no way of finding out who that individual is from that data. The only person will know it will be the foundation and the clinicians involved in the study. And we would have no other way of knowing it. So we did apply a grant. As I said, I'm an ally on a long haul. I'm not just blowing hot air here. So we did apply for an NIH grant last year with this plan. Unfortunately, as you know, with NIH and government funding, it didn't get funded. But again, N is equal to 1 is not, we'll keep on trying this. But as I was telling Marcy, it's also good for the foundation to get in there and sort of push the cases. The more government hears from the foundation, patient bodies to say this is required, is important. It sort of rises up to the top, because it's always there are many competing interests for limited pool of funding. And also, as the foundation gets more and more stronger and sort of attract more money, I mean, some of these projects can be kickstarted with small amount of money. You don't require millions of dollars to do this, believe me. A small amount of kickstart funding, anything would help get this started. But again, I am in for this long haul. This kind of things don't stop me. I haven't reached where I reached by stopping. I request Dr. Moore to send me if he has families like what I predicted to send. So I will use my own funding to, at least as a pilot, to see if it can be done. So he was very kind, he was very generous. He found us 2 families in Australia who is exactly the time I said we need. So the family on the left, the young man had both Mort Wilson as well as a long segment Herschbrand disease, severe. And I think the gentleman is deceased now, and I might be wrong, but it was a very severe case. And the young woman here on the right only had Mort Wilson syndrome. So this was like a test case for us. We asked the question, can we do this? If what I'm saying is correct, then there should be something different on the left and on the right, right? So we did whole genome sequencing with my friend and colleague, Dr. Tishel Turner at the Washington University and at NYU Genome Core here. Again, this was de-identified. As you can see, the numbers are called MWS 1.2, MWS 1.3. I have no idea where these individuals are, who they are. Dr. Mort knows. The first thing we discovered the ZEP2 mutation, the specific ZEP2 mutation both these kids have. And it was surprising to us, or maybe it's not surprising to you, these mutations have not been reported before in the catalog I found of ZEP. So there was 2 unique new mutations in these 2 kids. But that's great. So at least they had the ZEP2 mutations, which is what defines Mort Wilson. But we also found, this is a very preliminary data. We just got the data like last week. We also found in the kid who had both Mort Wilson and Hirschsprung disease, I'll explain this graph to you. Basically, this region of the genome, the blue is the sequencing of the patient who had Mort, Wilson, Hirschsprung, and the black is their parents. And this is basically you're sequencing every base pair and asking the question, how many copies of that base pair of nucleotide of DNA I find? As you can appreciate here, at this region from here to here, the blue goes over the black, which basically means this region is duplicated. So this patient has a region in his chromosome 13 or chromosome 8 where the whole DNA has been duplicated. Now, we don't know what it's causing. We don't know functionally. As I said, we just got the data last year, but clearly this is different from the patient who only had Mort Wilson. So these patients are sort of, in a way, it's a way in our hypothesis we made. So there are other changes of patients who have multiple disorders beyond just ZEP2. As I said, I don't know the biological consequence of this duplication. That's what we're going to find out now. We're going to do studies to see what this duplication does. And hopefully we'll have an answer, like some kind of explanation. But again, a few days of my life, which is really satisfying in the lab, is when we find these kind of things. It tells us that, we are on the right track. And I said, nothing stops me. I am determined to get to the bottom of this, right? And the foundation plays a very important role as collecting patients, giving us phenotypes. The more we get, as you can understand, it's a game of numbers. We will get better and better at this. Right. So I'm sorry I've taken up too much of your time. I will sort of end here. So my lab is just starting. So it's a very small lab. So most of the work is done by me or another technician in the lab, Lauren. She's very talented. My training with Aravind Chakrabarti's lab, where I did many of the early work I showed. And I'm extremely, extremely grateful to the foundation for not only trusting me, engaging with me, giving me a platform to do this kind of thing. We completely rely on patient advocacy groups and foundations like you for our research. We are just a small cog in the wheel. We help you in whatever we can. But Marcy here, Margaret Adams, and Seattle, Al, last year I talked to Al, Deborah, Catherine, have been very, very helpful in sort of, engaging, helping. And I really thank you for your time and for inviting me. And I hope this was helpful, and I would be more than happy to take any questions you have. I can stop sharing my screen.
Thank you. Oh, that was really very intriguing. Some of it, I'll probably watch the video a couple of times so I can understand it. The more that we have access to this information as people who are not scientists and things. I think it's real important that we at least have access to it and can get a little bit of information.
And you're more than free to email me whenever you like. I've given my emails across.
Oh yes, and thank you very much. So if anyone has any questions, you can put that in the chat box. But I have a few of my own questions. So at this point, the foundation, the Mort Wilson Syndrome Foundation, is working to kind of revive its patient registry and revamp it so we can, again, ask members of the community to provide their genetic information, other things, so that there's a repository for researchers. And I know that you talked about using some information from humans, and then also, some information in biosamples or with mice. So how important is it to you as a researcher that we have things like this patient registry and that members of the community agree to provide their information? How important is that for your research?
That is the bedrock. In human genetics, the biggest bottleneck is getting patient information or getting patient information in one place. And I mean, that's our starting point, right? We need to have an idea of how many patients we're dealing with. So all this hypothesis falls through if you come back and tell me after you make a very solid registry where you know exactly the phenotype, you know the patient, you know what kind of a genetic mutation if they have, and you tell me, well, Sumantra, there isn't enough Hirschsprung patients among us, right? There's all Mort Wilson. So then the part of my hypothesis falls apart, right? Which suggests that some of it cannot be done. So having a solid data set, and NIH likes it, having a backing of a data set which suggests this is the numbers, this is the phenotype, this is what we know, it sort of gives you confidence that the hypothesis is on the correct path. So it's everything. It is pretty much everything. So having a good database is the first step towards any human genetic research. The second step is what you mentioned, the biorepository. As you understand, getting tissue samples always, I understand, a challenge, and I never underappreciate the fact that how much effort patients and patient families go through to give us samples, and so it is the more we collect, the more our power of detection improves, the more our validation sort of power improves, right? The mouse is after all a model, we model a lot of things, but then ultimately, if I find something which is not really happening in the human, it is not really informative to you. It can be great if we write a paper, academic interest, but it doesn't. So the biorepository, but I would say first, in the order of preference, I would first rather have a very good patient registry where again, as I said, I don't need to know their names, social security number, that can be yours.
Right.
But in terms of your, like what the status is, what they have, and if they have been genetic tested, what kind of phenotype they have. I mean, that is the first step. And that sort of gives us power to also argue that this is important. And then
right. Well, we're working on it, and we keep saying hopefully next month.
That's absolutely fine.
Hopefully by the first of the year, we'll be able to start inviting members of the community to share their information in the registry. And then the next thing will be the biorepository. And, that's kind of one of the reasons that I felt it was important to ask you researchers to come and introduce yourself. Because I know I've heard from some families, they've sent biosamples, they've given their information, and they never really hear anything back. What happened, what was done. And so we're trying to connect the circle back. So researchers know what we have available, where we are in the process, and our community understands why it's important.
And I agree that trust is very important, because we have a bad rap sometimes. I agree. I mean, it's not only me, people do take samples, right? And then you never hear back from the lab, right? The lab, there are reasons. So I think you can talk to Dr. Moore independently. You don't have to ask me. For every sample, he sent us 6 samples. At every step, I informed him and Dr. Wilson, this is where we are. This is the DNA we got. This is where sequencing is. This is where we've reached. So every step, at least I can speak for myself, you would hear from, I would not disappear. Unless you will hear from me where we are at, and sometimes, experiments do fail. So we will also hear from that and say, look, we tried, but unfortunately, DNA wasn't good. I mean, so there would be, everything would be transparent. And one thing we can plan, again, I don't want to take up too much time today, is some kind of a common shared registry where we put up data and you have access to it. It's a complete transparency. You have a timestamp, a date, where the data came from, who generated it. So you can go back and forth and exactly who is.
Right. I think we're somewhat limited by the company that hosts the, because we don't host these as a foundation. We work with other organizations, and they are also, charged with all of the data security and those kinds of things, which that's one thing that we also need to learn more about. What are the measures they're taking to making sure that everyone's information and data is secure. So I have another question here. What you've talked about is a lot of information about the genes, the genotype and the phenotype, how they kind of work together, and all of the nerves in our brain and all of those in our gut, and how they're very connected, and it's very complex, and that you're learning a lot more, it sounds like, in almost every research about what that means and the different things that impact maybe the level of Hirschsprung's disease that an individual might have, like in the future when individuals can get genetic testing readily and quickly, and it's not very expensive, then it might help clinicians be able to understand better how to treat. But now, as we are, kind of how do you see your research that you've done and what you've got planned in the future? How do you think that can impact the treatment of Hirschsprung's disease and things? Even if we're looking into the future, kind of how do you see what you do affecting more clinical treatment that will, that's something that, families are really looking at and wanting help with.
Right. So I will start off by the caveat that we are far away from it yet. So don't take my word for Sumantra can do it tomorrow, or somebody. So that's a caveat, just as a thing to look. But it's a very good question. So the first biggest challenge for any congenital disorder, as you can appreciate it, it's all the process already happened while you were in the womb, right? When we see the child or the baby, we are seeing the end result of that change. You are not seeing it in action. Like a adult disorder, like you get cancer, you see it in action while you're adult. So we can only sort of go in there once the process is over, once you're born. that being said, what these kind of research tells you, as I told you in the mouse model, is, I mean, what kind of cells are missing? So do we need to actually go back and repair every cell type in the gut? No. Can I repair only this 10% cells which are missing? Right? I will never, so you'll never become like a 100% normal gut. But can I push the kid just over the limit of expression where now these cells can start doing a little bit of the function? So it improves the quality of life, right? So you find out the cells which are missing, you try to regenerate them in the lab. So now we are working with Lawrence Truder's lab here in Mount Sloan Kettering to try to make ENS from like iPSCs to see, because it's the biggest bottleneck. People have made every different kind of cell except the entry neurons. Those cells are a little difficult to generate, but they have got a protocol working now, at least in a lab. In a lab setting, in a dish, we can start making these cells, right? So if I know what cells are missing, I can know what cells to create. And then hopefully one day we can go back into the patient and replant those cells in the gut, even those cells have, like, even if the gut is now fully formed, right? And hopefully those cells will now recolonize, at least partially recolonize. So you will improve the quality of life. You will not make the kid completely like free of like aganglionotic phenotypes, but at least from going completely like dysmotility of the bowel, having a colostomy bag all your life, you now start making interventions where you say, we've repaired, I apologize for the word repair, I don't know if it's a correct even word, but at least we have attempted to change some of the phenotype of that in that gut. But we can't randomly go and put random cells in there, right? This is not a mouse. So it's a human being. So we need to know what cells we're putting in. And in some cases, we might even tell you an answer that, unfortunately, this mutation is so severe, the cells are so gone that there's nothing you can do. And I think for a parent, sometimes even knowing that information is helpful. Like, even if it's a negative information, some information is better than no information. Right. But in some cases, we can tell you, there is a chance. There is these cells we know are the ones which are missing, and we can now grow these cells in a dish. So hopefully we can now transplant those cells back, and those cells will basically go in there and sort of partially colonize your gut. So that's a hope.
It is hope. And, information is power. We all know that. And I think from especially new families who are newly diagnosed, a lot of what I hear is just fear of the unknown, because there are a lot of things they don't know. But as we get more families sharing information, just their stories, I think gives people comfort. So one other kind of question, in the beginning, when you were talking about Herschbrand, you talked about how it seems to appear in males more than females. And then you also talked about how with individuals with Mott-Wilson syndrome, a lot more individuals are affected by Hirschsprung's than the general population, kind of like instead of 1 in every 5,000, that's about 50%, I think you said. So do you find the same kind of trends as far as male and female individuals, or not so much?
So people who have Hirschsprung and Mott-Wilson, there is a sex bias. It's, I think, 2 to 1. So people, that's why the patient I showed you who had both, it was a male. So there are more, but people who just have more, if you just look at only Mott-Wilson alone, who don't have Hirschsprung, I think the sex difference, there's no sex difference. Male, female can be
okay. Interesting.
But the other thing to realize that it's a rare, the numbers are small, right? I mean, I think the numbers we've looked up so far in these papers are like, I think total of 600 in overall worldwide. So out of which like a proportion of them are Hirschsprung. So if you remove the Hirschsprung and Mott-Wilson patient, the only Mott-Wilson, there is no sex difference. The sex difference, I think, is derived patient who have Hirschsprung. So there are more males there than females, like just normal Hirschsprung.
Okay, very interesting. Yeah. So I really appreciate all of wonderful information for taking time out of your day. One final question.
Yes, absolutely.
So I was reading some things about researchers and where sometimes they want to connect more with families and show kind of their space and their lab and kind of what that looks like. Explain. I know we might not be able to really relate to everything or understand everything. Kind of, would maybe you consider sometime in the future doing a virtual tour of your lab so we can kind of see where you work and where all this important stuff? That would be a fun thing to do, so we can learn a little bit more about what goes on behind the scenes.
I'm more than happy to. I'm always open to science communication, because sometimes I think as a scientist we fail to communicate to the wider audience what we are doing. And for a family to get a glimpse of where this work is done, there's no mystery in it, right? Anybody can do it. I always tell this to people. There's no mystery. In the movies, we make it like seem like there's some kind of mysterious world we inhabit, like, we're wearing lab coats and pouring colorful solutions in. I mean, it's a very, very important thing to understand, like to open the doors to a community and show them that this is what I will more than happy to. I mean, my
That would
lab
be a
doors.
fun thing, I think, for us as people
Absolutely.
who don't get to see the inside of the lab, and it seems a little abstract and like
Absolutely.
PowerPoint, but it would be kind of fun just to see
Absolutely.
you in your real environment there
Absolutely.
and all the things that you do. Again, I just want to say, Sumantra, thank you so much for taking the time today. Right before the call, we did talk a little bit about the fact that the Mott-Wilson Syndrome Foundation is working right now to develop some processes to fund some research on MWS, and also kind of connecting the circle, getting more involved with researchers and helping to create some collaborations there, and also connecting the researchers to our community and our community to the researchers. So we have a lot more open communication, more transparency in research.
Absolutely.
Samples are being used and where funding is or where it's difficult to have funding. So I just thank you again. Thank you for all of those who attended today and for all of those who are viewing the video later. And just really appreciate you. If you have any additional questions, you can always go to the Mott-Wilson Syndrome Foundation website and contact us there, and we can be sure to get you as much information as we can. Again, thank you all. Thank you again.
Thank you, Marcy.
I mean, I really appreciate it. Look forward to maybe connecting sometime after.
Absolutely.
Here, getting a tour of your lab. That's
absolutely
like a fun thing to do. All right. Well, thank you again. Have a wonderful day, and best of luck to you in all your research. And I just want to say thank you for choosing to become a researcher. I know it's not an easy path, and it's certainly not a very lucrative one for most people. But you are making a difference. You know, we're learning more, and as with most things science, it's the future that matters, and where all these things matter. So thank you very much.
Thank you for having me.
Thank you to those who attended. Everyone have a wonderful day. Thanks again.
Thank you.